Well, good evening, everyone, and welcome to the marvellous surroundings of the Sheldonian Theatre. It's great to see so many of you here for the Centre for Personalised Medicine's annual lecture. So my name is Annika Lucassen, and it's my privilege to be director of the Centre for Personalised Medicine, or CPM. And I just wanted to tell you a little bit more about the Centre for those of you who don't know about it. And I'm just polling the front row here. Some of you haven't heard of it before introducing tonight's speaker. And hopefully that will also show you what a great choice tonight's speaker is to be an annual lecture for us. So the CPM is a multidisciplinary scholarship and engagement vehicle to enable a wide range of audiences to researchers, clinicians, policymakers, and the public to explore the benefits, but also the challenges of attempts to personalise medicine. So I myself am a clinical academic, a clinician interested in translating the massive technological and scientific developments we have seen over the past 20 years to what they mean or don't mean for individuals, families, or other groups. And I'm also interested in researching how to do so in a setting of often deterministic expectations of data, particularly of genetic or genomic data. I am, of course, also a member of the public and a potential patient, just speaking to how our audiences overlap quite significantly. The CPM is one of those great Oxford ideas. It brings together the interdisciplinary academic culture within a college, St Anne's College, with the research rigour of the Nuffield Department of Medicine, specifically the Centre for Human Genetics within that department. What I really love about the partnership is that it allows nuanced debate of complex topics, often ones where opinions can start off quite polarised. Through debate and a wide range of activities and events, we hope to advance the narrative around what scientific and technological advances can deliver now, what they might deliver in the future, what are the hurdles in the way, and where do we need to be more realistic in our expectations. The CPM is funded in large part by a very generous philanthropic donation, the Stanley Ho Medical Development Foundation, for which we are extremely grateful. Do have a look at our website if you want to know more about us, and do get in touch if you'd like to collaborate in any way. So this evening, we are really delighted to welcome Philip Ball to this year's annual lecture. Philip is a freelance writer and broadcaster, trained as a chemist at the University of Oxford and as a physicist at the University of Bristol. He was editor of the journal Nature for more than 20 years and writes regularly in scientific and popular media, and has written many books on the interactions between sciences, the arts, and wider culture. I first came across your work through the book The Music Instinct, which I loved, although I realise you have many other works, and I think many, if not most of them, have won prizes. As you explained so well in your most recent work, How Life Works, the ways in which we are taught to think about DNA or genes can sometimes be unhelpfully simplistic. And as we move towards increasing the use of genomic and other data to inform healthcare and medicine more generally, we need to better recognise the complexities in order for personalised medicine to flourish. I'm really looking forward to hearing your thoughts on how things might need to change to create an environment where we're all better equipped to access the benefits of medical care involved by genomic or similar data without the risk of being blindsided by its limitations. We hope to have some time for questions at the end. Philip, I'm going to hand over to you. The floor is yours to talk about how life works, dealing with biology's changing narrative.
Well, thank you, Annika, and thank you to the CPM for doing me the honour of asking me to give this talk, which is, of course, particularly delightful in surroundings like this. It might be a bit chilly, but that's the price you pay for grandness, particularly in Oxford, I guess. I want to start here. Last month, This paper was published by a team of researchers from Melbourne and from here in Oxford. It was a paper in Nature, and it argued that what is known as polygenic genome editing could offer significant health benefits at the population level. And the idea that they explored is that as we can now identify the gene variants that in the population that are associated with common diseases like coronary disease, schizophrenia, Alzheimer's, type 2 diabetes, we can imagine screening IVF embryos to look for the presence of those gene variants in their genomes, and then use gene editing technology to snip out the risky variants and replace them with healthy versions. And this, of course, sounds like the ideal sort of personalized medicine that was first proposed when human genome sequencing began, to tailor each person's DNA so that it doesn't contain gene variants that would put them at risk of serious diseases. And the authors of the paper made this sound like an extremely promising idea. For example, they said that by screening and, where necessary, replacing up to 10 gene variants linked to type 2 diabetes, it might be possible to reduce the prevalence of that condition in the population about 50-fold. So 50 times less diabetes. Now, I should make it clear that these predictions weren't based on experiments. The technologies do exist for editing human genomes, including those of embryos. But actually doing that in the way that the researchers described in this paper would be illegal in this country and in most others, but partly because we don't know how safe it would be, but also for other ethical reasons. So the predictions were based on mathematical models, given what we know about the effects of certain gene variants on certain diseases. So all this might sound very promising, but alongside this paper, Nature took the highly unusual step of publishing a commentary saying, in effect, that we shouldn't believe it. The authors of the commentary said that the whole idea was unsafe and unproven. So, what's going on here? Who are we meant to believe? Well, I'll come back to that question and this work later on, the question of what the work is really about and who I think is right. Although a spoiler is that I think the critics are right. But I'm starting with this story because, to me, it illustrated the central point that I want to make in this talk. The two sides of this debate, to me, exemplify the two different notions of how life works, or specifically what role genes play in that question. I would argue that the idea of polygenic gene editing is predicated on the traditional view of the genome, the DNA in every one of our cells, a view that regards it as an instruction book. So if some of those instructions are faulty, giving rise to a malfunctioning organism in some way, then we can use gene editing techniques to rewrite and correct that faulty or those faulty instructions. And the authors of the Nature paper pointed out that there are some complications with that approach, for sure, but those are secondary problems, and we will hopefully figure out how to address them in time. The critics, on the other hand, are really saying that those complications are fundamental to what genomes do, and that in fact the instruction book picture is the wrong one. So on the one hand, we have a simple picture of biology that in reality gets a bit messy because biology is like that. On the other hand, and this is what I want to suggest to you tonight, the complexity is absolutely fundamental. And the simple stories that we've tended to tell so far can be profoundly misleading. And if that's the case, if we have misleading stories that end up with advocacy of large-scale gene editing of human embryos whose outcome can't really be predicted, but which would in any case be inherited by future generations, then it seems pretty urgent that we have a conversation now about these different views of how life works. Well, starting with the idea that it's actually all very complex doesn't sound very appealing, does it? How is that going to help any non-experts understand biology? I'm not going to pretend that everything that I have to say will be easy to take in straight away, but my contention is that even complex things like us work according to rules that we can begin to grasp. And that sometimes a story only might seem complicated because we've started it in the wrong place. Now, I'm anticipating, in fact, I'm pretty confident now that this audience will have diverse backgrounds, and some of you might already be struggling to figure out what it is that I've said. What are genes exactly? And here's a secret, biologists are no longer sure what the answer to that question is. What do I mean by gene variants? What is DNA even? How do you edit it? And how come some genes seem to be associated with diseases? So let's go back to what's often regarded as the start of modern genetics and modern molecular biology, the discovery of this structure of what has become really a cultural icon, this double helical structure of DNA that was deduced in 1953 by James Watson and Francis Crick, drawing in particular on studies of DNA crystals made by Rosalind Franklin and her co-workers. And at that time, DNA was generally thought, not universally, but generally thought to be where the genetic information that is passed on between generations, where it is stored. And in this double helical structure, Crick and Watson could see how it is that our DNA is able to encode that genetic information, because each of these two strands is made up of a string of just 4 different chemical building blocks, which we denote by the letters, the first letters of their names, C, G, A, and T. And this was said to supply a means of encoding information rather like, just like really, the strings of ones and zeros in binary code that was then starting to be used to encode information on magnetic tape for digital computers. So the idea is each one of our genes is a short segment of that code, and we have many thousands of these genes in this string of letters that for us, for we humans, is 3 billion letters long, and which we call the genome. So here then was apparently how we work. The single fertilized egg from which we all begin comes with a set of instructions in its genome, and all that needs then to happen is that those instructions get read out to build us. And so it seemed now that the task was simply to read those instructions, and that was what was done in the Human Genome Project, which began in 1990 and was completed, at least as a rough first draft, by the turn of the millennium. And so here it is, our instruction booklet, or our blueprint as it's sometimes called, all the letters of the human genome, which you can actually go and read, should you be inclined, in these 109 books held in the Wellcome Collection in London. So is this the secret, then, of how life works? Well, it's clear that actually DNA doesn't do anything by itself. If you put a human genome in a test tube, you can throw at it all the other ingredients that life requires, amino acids and lipids and all the rest of it, and nothing will happen. You certainly won't get a human being out of it. You won't even get a single cell out of it. For life to happen, the genome needs to go into an already living system, at the very least into a cell. And so we're left with this rather unsatisfactory picture. You can call DNA the secret of life so long as you just add life to it. But in the blueprint image, this isn't really the point. DNA, according to that picture, contains all the information you need, and the cell is just a readout mechanism. And the picture we're often given is then something like this. So you have the genes, and then something very complex happens to them in that black box, and out come organisms. And what goes on inside that black box is so horrendously complicated that it's sometimes said it's best that you don't even bother about it, don't even look, but rest assured that scientists are sorting it out and that one day we'll have it all straight. However, that advice neglects this inconvenient fact. Genes are very definitely not alive. Organisms certainly are. And so it seems that this is where life happens. So I'm afraid that to understand how life works, we really do need to peek inside this black box. And I'm hoping to give you a glimpse of it. And in all its glory, it is mind-bogglingly complex. But I hope to persuade you that we can at least make some sense of it. And what's more, that in the 2 decades or so since the completion of the Human Genome Project, the sense that we are starting to make tells a rather different picture to the one we've traditionally been told in the genomic era. Now, Francis Crick started to open up that black box a little, and this is what he discerned inside it. This is what, in the late 1950s, Crick called the central dogma of molecular biology. And I think it's a label that is misleading in the same way that the Holy Roman Empire is sometimes said to be misleading, because that wasn't really holy or Roman or really an empire. And similarly, the central dogma is not really a dogma. In fact, Crick admitted that he used that word without really knowing what it meant, but it's not really so central either. Well, let's take a look at it. It sums up what was already believed at that time and what was soon proved to be the case, that what the genes in DNA really encode is instructions for making these molecules called proteins. And in the usual cliche, proteins are the workhorses of the cell. They are the molecules that help the cell's biochemical reactions happen, generally by acting as catalysts that help to convert one biological molecule into another. And the readout of this code is in this two-step process via this molecule called RNA. And so this is in detail what happens. At first, the code in a given gene is copied, the technical word is transcribed, into this molecule called RNA, which is very similar to DNA. So just that little bit of the genome is transcribed, and this piece of messenger of RNA, so-called messenger RNA, then goes off and is seized by a piece of molecular machinery called the ribosome, which uses the information in the RNA to put together a particular protein, a string of amino acids in a particular sequence that crumples up into a particular shape. And that process of making the protein is called translation. And there are thousands and thousands of these proteins in every one of our cells, and they're often portrayed as little molecular machines that put us together. So here is how that idea would be expressed in the context of the central dogma. And this fancy word phenotype here just means all the traits we have, how tall we are, what form, what shape we are, what color our eyes are, what color our skin is, even what behaviors we show. Basically, the phenotype is our individual selves. And in this picture, it all comes from what is encoded in the DNA, from our genotype. Now, it turns out that the DNA sequence encoding a protein can be varied a little bit. You can substitute one nucleotide for another, one of these letters, these chemical letters for another in some places, and you still get basically the same protein that does the same job. And these are mutations, these substitutions, these are mutations that happen over the course of evolution. And so it results in there being lots of slight variations, lots of slight variants of each gene throughout the population. We all have different variations of that. The technical term for those variants is alleles. But some variants make proteins that don't work as well, or perhaps don't work at all. So, for example, some alleles of a gene called HBB, which encodes a part of the hemoglobin protein that carries oxygen around in red blood cells, some of those variants make a protein too faulty to work properly. And the result of that is that the red blood cells become misshapen. And that is the cause of sickle cell disease, which typically can be an excruciatingly painful disease and often results in early death. And it's now becoming possible to treat this condition by gene editing. And that involves taking out the blood-forming stem cells from the bone marrow of the patient and replacing the faulty HBB gene variant with a healthy version using a gene editing technique known as CRISPR. And you can think of CRISPR as being, it's a bit like a pair of molecular scissors that can be programmed to find a particular sequence in DNA and then snip out that segment, and then you can substitute a different segment back into the DNA. And then those edited stem cells are infused back into the patient and they start to produce proper functioning hemoglobin. And this method has been trialed, was trialed some time ago in a few patients. And at the end of 2023, a therapy using CRISPR was approved for use in the UK and in the US. And you might have actually heard just a few weeks ago that such a gene therapy treatment for sickle cell disease was approved for use on the NHS. So evidently, gene editing can be used successfully to treat genetically based diseases. So what's wrong with the idea of editing many genes that way, if that's what's called for to cure some diseases? Well, let's go back to Crick's central dogma. What many biologists took away from it was the idea that all the information flow in biology is from the genome, from our so-called molecular instruction book, upwards to the whole organism. And each gene according to this picture, encodes a protein, and those proteins somehow create our tissues and our bodies and our traits, including our propensity to genetically related diseases. But if we try to go from genotype to phenotype, things get messy. Many genes that seem to influence one trait turn out also to influence others. And the technical term for this is pleiotropy. And it was once thought that pleiotropy was just a quirk of a few genes, but we now know that actually it's the norm, that most of our genes influence more than one trait. And some studies suggest that on average, typically a gene will influence maybe half a dozen different traits in some way. What's more, any given trait tends to be affected by several, perhaps many genes. Traits are polygenic. So complex traits like intelligence, susceptibility to heart disease, even height, are typically influenced by tens or even hundreds of genes. And some researchers have concluded that many of our traits aren't just polygenic, they're omnigenic, meaning that there is no gene that doesn't influence to some degree that trait, even though most of those influences are very, very small. So a disease like sickle cell disease that seems influenced by only one gene is the exception. And yet genetics is typically taught with reference to this sort of relatively straightforward genetic relationship, not because it reflects what is typical, but because it's then quite easy to understand how conditions like that are inherited. I suspect many people will remember those Punnett squares that you're taught about to understand that sort of inheritance. Well, this looks pretty messy, but it's worse, because we can't even say in general that this gene variant is good and that gene variant is bad. The effects that a variant has on the phenotype typically depend on what other gene variants are present too, on the overall background of genes throughout the genome. Even for some single gene diseases, sometimes someone with the bad gene might not show that disease at all, or might only show it mildly because the gene's effects have been masked or compensated for by other genes in that genome. And those effects might also depend on lifestyle choices. So genes are anything but deterministic of particular traits, and we can't, in general, predict the effects of one variant without taking into account all the other variants in the genome. So you might see then why, even if gene editing was 100% accurate, which it isn't, editing many genes in the hope of engineering particular traits, say lowering the risks of a polygenic disease, can be something of a lottery. Not only can we be not sure how it will turn out for that condition, but it may be that by altering a variant to suppress one disease risk, we may be increasing the risk of some other disease. And that is not a complication of how genes work. It seems to be a fundamental aspect of genomic complexity. And it's because of this complexity that this idea that I mentioned at the beginning of polygenic gene editing to improve the health of the population, and incidentally, that's exactly what it sounds like. That is a eugenic notion in the literal sense. It doesn't seem to be feasible. The proposal seems, in general, doomed to fail for the same reason that Francis Crick's own enthusiasm for eugenics was mistaken, which is that Crick's central dogma is not the story of how we go from genes to the complexity of an organism like us. So where does all that complexity come from? That's the big question, really, and it's one that's still far from understood. But I want to show you a few examples of how that story has changed since Crick introduced the central dogma, and in particular, since the human genome was completed. One of the surprises of the Human Genome Project was that we turn out to have only 20,000 protein coding genes. That is about as many as a tiny soil-dwelling worm called the nematode, and it's barely more than half of what the banana plant has. So, what's going on there? And incidentally, in this diagram, I've shown what was estimated as the gene count before the Genome Project began. Typically, they were figures like this, 80,000 to 100,000, because we're more complex, right? So it stands to reason we must have more genes. That's clearly not the case. Here's a second complication. In the 1990s, there were one or two genes for which biologists didn't seem to be able to find the corresponding proteins. And in the end, they had to conclude that that's because there aren't any. Those genes simply make RNA, and not the messenger RNA that gets translated by the ribosome into a protein. Instead, that RNA has some biochemical function in itself. It does the kind of thing that we thought proteins do. And these RNA encoding genes are called non-coding genes, not evidently because they don't encode anything, but because they don't encode proteins, and that's what we thought genes did. So there were a few of these non-coding genes. Well, okay, biology is always full of exceptions, right? Except that this wasn't an exception. Over the past 2 decades, the number of these non-coding genes has been creeping up. And exactly how many of them there are is contentious because it can be quite hard to figure out if they have a real function or not. But according to some of the best figures that we have, this is what has been happening. And some years back, the number of these non-coding genes overtook the number that code for proteins. What's more, some current estimates say that there are going to be lots more of these non-coding genes discovered in the future, and they're going to turn out to vastly outnumber protein coding genes. And there are lots of different families of these non-coding RNA molecules. Some are quite short. They might just be maybe as few as sort of 20 or so of these chemical letters long. Some are much longer, hundreds long. So there are all sorts of families of them doing different things. So you can see that the genome isn't really about what we thought it was about. And yet somehow we still seem to be telling the same story about it that we were telling back in the 1990s, or even actually back in the 1970s, and that doesn't seem right. Well, what about the genes that do make proteins? It was initially thought that for each protein-coding gene, there's a corresponding protein, which sort of stands to reason, but that's not quite right. Each of our genes can be used to make several different proteins, on average around 6, but in some cases dozens or even hundreds of proteins, not all of which might seem to do anything useful in the cell. So we have many more proteins than we have protein-coding genes. Actually, no one really knows exactly how many proteins we have. How is this possible? Well, it's because, as was first discovered in the 1970s, the messenger RNA transcribed from a gene is typically edited before it gets translated. So another piece of molecular machinery called the spliceosome chops up the RNA, throws away some pieces called introns, and stitches back the fragments called exons in different orders. And what decides how that editing and how that splicing occurs is typically the context, is typically information coming from higher levels, from the state that the cell is in, for example. So a gene might produce one protein in one type of tissue and a different protein in another. And in some cases, these alternatively spliced proteins, in some cases, they might work in slightly different ways. They might do slightly different things. In other cases, they might do dramatically different things. So take, for example, the proteins called NCAMs, and that stands for neural cell adhesion molecule, because these are proteins that influence the way nerve cells stick to each other. So there's one gene in the human genome that makes a whole variety of NCAM proteins, depending on how its 19 exons are put back together. And in theory, there are thousands of ways of doing that. And they have very different functions. Generally, they're connected to the formation of synapses where neurons, if you like, speak to one another. So there are some forms of NCAM protein that help a synaptic junction in our brains stay stable. So they help us learn stable patterns of behavior. There are other NCAMs coming from the same gene that make a synapse relatively loose and liable to be rewired to give us neural plasticity. So here we have virtually opposite phenotypic consequences coming from the same gene. What's more, NCAMs also affect interactions between other types of cell, not just nerve cells, but cells in the immune system. So what exactly should we say it is that the NCAM gene does for the phenotype? I think the truth is that all we can really say is that it provides a resource. It provides molecules for controlling interactions between cells that the organism can use in a variety of ways depending on the context. It's not an instruction for anything really as such. I mean, I like to imagine it a bit like the genome saying to the cell, here's a molecule that you can use, you can stick it in a cell membrane, and it'll influence how the cell responds to other cells around it. Now, you decide what to do with it. In other words, the information flow here isn't, as the central dogma implied, all from the bottom up, from DNA to RNA to proteins. Some crucial information for determining which protein we get is coming from outside that system. And this is just one of the ways in which, in order to build us and to keep us alive all these years, information doesn't just flow up from the genes to higher levels of organization. It also flows down and sideways and in and out and always. It is an open informational system, not a closed one. Now, as I say, this alternative splicing of proteins has been known about for a long time, since the 1970s, but there's been another, more recent shake-up in how we think about proteins. Here's a snapshot of a human cell, and this isn't a schematic. It's computer-generated from actual data taken from a real cell, and you can see it's insane. It's madly crowded. But no matter, the usual story goes, because each protein has a very particular shape, which will fit like a lock into the key of the molecule that it's meant to transform. And so it will only recognize and work on that molecule and ignore all the others. Proteins have very specific shapes in order to do that. Here is one of them. This is actually the hemoglobin molecule that I mentioned earlier. Over the past 2 decades or so, it's become clear that many of our proteins, including some with the most important jobs in our cells, don't work this way. They don't look like this because they don't actually have a well-defined shape and structure. That structure is left loose and floppy, or as biochemists say, intrinsically disordered. And this isn't some failure of the genome to give the proteins a proper shape. This is a deliberate feature that evolution has, so to speak, chosen, because there's actually much less of this intrinsic disorder among the proteins of simpler organisms like bacteria. So it seems that evolution has found it useful, perhaps even necessary, to have disorder in proteins of complex, multicellular, multi-tissue organisms like us. And the more complex an organism is, the more disorder there is among its proteins. Proteins with intrinsic disorder tend to be far less choosy about which other molecules they stick to. They're rather promiscuous in their molecular unions. So this old idea that the molecular chaos of the cell is kept orderly because each protein is highly selective about what it interacts with doesn't seem to work in general. And that's well illustrated by this protein with some intrinsically disordered regions. It's a protein produced from the gene called BRCA1, probably one of the best known genes as these things go, because certain variants of this gene are associated with an elevated risk of breast cancer. So much so that this name, BRCA1, it basically means the breast cancer gene. That's how it's known, which is obviously an absurd thing to call it, because evolution isn't going to give us a breast cancer gene. The truth is that, of course, this protein, when it's functioning properly, does all sorts of functions in the cell. In fact, it does so many functions, it's very hard to say exactly what this protein is for, if you like. Because of this intrinsic disorder, it is able to interact with a whole bunch of other molecules in ways that are generally connected to what is known as gene regulation. And that's what I want to come on to next, because that is really the big deal in how we work. You see, the standard argument for how we get to be so complex with so few genes is that the complexity arises from the very tangled network of interactions between our biomolecules. And in particular, this molecular crosstalk is invoked to explain how it is that different genes are turned on and off in different types of cell, making, say, a heart muscle cell different from a skin cell or a liver cell. All of them have the same genome, but the idea is that the genes and the proteins that are made from them can talk to each other so that one gene can alter the activity, the level of transcription of another gene. This notion of gene regulation is also old. It's been around since the 1960s, when the French biologists François Jacob and Jacques Monod showed how it worked for one particular process in the bacterium E. coli. So E. coli can digest two types of sugar, glucose or lactose, but it's not very efficient for the bacteria to be making both of those enzymes needed for digestion if there's only one of those sugars around. So there's a switch, which Monod and Jacob called the lac operon, a switch for turning on and off the production, the expression that is, of the so-called lac enzymes that digest lactose. And this is basically how it works. In short, there's a protein, this green thing here, that can recognize and stick to a short segment of DNA just next to and just before the lac genes. And when it sticks like that, it blocks the molecule called RNA polymerase that will otherwise come down onto the DNA and start the transcription process for making those enzymes. So this green protein, it's called a repressor protein. But if there's lactose around, the lactose itself will bind to that protein and change its shape in a way that means it can no longer stick to that place on DNA. So off it comes, and the transcription can then proceed, and the lac enzymes can be made. So it's kind of like a digital switch. It's either on or off, and it allows one gene to regulate another. And for a long time, molecular biologists imagined that gene regulation in organisms like us follows this same sort of simple logic, effectively wiring our genes into a kind of digital circuit like those in microelectronic devices. Well, you can probably guess what I'm going to say. That's not how it turned out. It's not as simple as that. Very often, gene regulation in large, complex creatures like us is much more complicated. It still involves proteins that interact with parts of DNA to control gene expression. These are proteins called transcription factors, but there are a whole bunch of other things that are involved as well. There are these regions right next to the gene that somehow control or influence that process, but there are other regions that seem to influence gene expression that are miles away on the same chromosome. And that somehow have to be brought close to the DNA that they're regulating. These are regions called enhancers. And some of those enhancers, it turns out, encode some of this non-coding RNA that I mentioned earlier. So in us, gene regulation involves a whole bunch of components. These transcription factors, regulatory RNAs, these pieces of DNA that have to be brought close to the gene they regulate by pulling out big looping strands, like sort of pulling out a bit from a tangled ball of wool. And all these components, they don't fit together in some kind of precise gigantic regulatory assembly. They just form a loose, dense cluster, a kind of liquid drop, a bit like the blobs of vinegar in the oil of salad dressing. So for us, gene regulation seems to be done by these loose committees of molecules, all talking to one another rather indiscriminately in a kind of analog fashion. And it's amazing, really, that somehow all of these components combine to make a reliable decision about whether to switch on or off a particular gene, despite the fuzziness of all these interactions. And in fact, blobs like this called biomolecular condensates and made up of mixtures of many different kinds of molecules, seem to be involved in all kinds of other cell processes too. For example, they help our cells survive stresses like heat. They help cells repair damage to DNA. Typically, they're made from proteins, and in particular from those disordered proteins that I talked about earlier, and often RNA, particularly that non-coding RNA that I talked about earlier. And these biomolecular condensates seem to be a key feature of how cells organize and compartmentalize their molecular components. And to find out more about that, you can read an article in this month's Scientific American, which I wrote about them. This sort of fuzziness that we see here and elsewhere in molecular interactions, and the way that it produces collective decisions rather than simple digital logic based on this molecule talking to that molecule, this is a characteristic feature of our own molecular biology. And there's still a lot of it that we don't understand. But I think we can start to say why it is that for us, this kind of fuzzy, analog and rather open-ended way of molecules working makes sense. Because when you come to think about it, the more complex an organism is, the more this blueprint or genetic instruction book looks like a terrible way to do things. If working correctly depended on each of those instructions being executed perfectly, as if in some fantastically piece of clockwork mechanism where everything has to be in the right place at the right time, that's just not going to happen. Not least because the molecular world is full of randomness and noise. It'd be like expecting a mechanism like this to go on working if we shoved it in a warm bath and shook it around. To avoid the fragility that comes from relying on everything being in the right place and working perfectly in the right time, you need totally different design principles. And that's surely why instead we have these fuzzy molecular mechanisms where the details often don't really matter. These molecular committees seem able still to come to good decisions even if some of their members are not there or are not working properly. And what these principles are really about, I think, is about taking the responsibility for the correct functioning off the lowest levels of the system, the molecules and the genes, and handing it to a higher level. In other words, we work in a way that is specifically designed to take the pressure off our genes, to make them, in general, no longer the real cause of our traits and behaviors, of our phenotype. Mistakes and malfunctions at the lowest level can then be compensated for higher up. And this is something that I've suggested calling causal spreading. Now, I don't think there is a single machine that we make that works like this, which is why I think it's deeply misleading to speak of the human as some kind of soft, wet machine, let alone a machine made by genes. Whatever metaphors we want to use for speaking about how life works, they shouldn't be metaphors to do with clockwork or computation. What organisms kind of seem to do instead might be regarded as a kind of cognition. And this is what the biologist Mike Levin and the philosopher Dan Dennett have argued. Not in the sense that organisms are somehow, or cells are somehow aware of themselves, but that we can think of cognition at least as a better metaphor for how they work. And what really distinguishes living things from any machines that we've yet made are that we are not automata, that living systems have agency. And by that, I mean that they're able to manipulate themselves and their environment in order to try to attain some self-determined goals. And I think that displaying agency is a much more fruitful and general way to think about what living systems do and what they are than to try to compile some checklist of, what life has to evolve, like reproduction and metabolism and homeostasis and so on. That's why I think to have an overarching view of how life works, I think that biology needs to develop an understanding and hopefully a theory of agency. We don't yet know how to do that, although I'd recommend this recent book by the neuroscientist Kevin Mitchell that offers a great start to that conversation. Now, this new view of how life works matters particularly when it isn't working so well, when we want to put it right. It matters for medicine. And this issue of causal spreading matters in medicine because in order to effect some change in the outcome of a system, we do best to intervene where that outcome is actually caused. Is the cause of such and such a disease some gene that we should be targeting, say? Well, in the case of something like sickle cell disease, it does seem to be. But for most diseases which are polygenic, that's not clear. Here, I think the old debate about reductionism versus holism becomes irrelevant. In between those two extremes, there's a whole hierarchy of other levels, and the question is about finding the right level of intervention. And there's plenty of reason to suppose that the right level is often not the genes. Take cancer, for example. Cancers can arise because of mutations in our genes, maybe because we get old or because we've been exposed to things in the environment that cause those mutations. So it used to be thought that we could find cures by looking at the genetic roots of cancer. But those efforts have turned out to be largely disappointing. And that it seems that the most effective level of intervention is typically a higher one. One in particular that's now attracting a lot of attention is intervening at the level of the immune system. As cancer biologist Michael Yaffe said in 2013, we spent fruitless years looking for cancer-linked genes, not because we thought that they were the key to developing new treatments, so much as because we had the techniques for looking for them. So this is what he said. Like data junkies, we continue to look to genome sequencing when the really clinically useful information may lie someplace else. And the complexity of the causal relation between genes and cancer is illustrated by the BRCA1 gene that I mentioned earlier. Is it right to say that particular variants of BRCA1 cause breast cancer? Not all women who have those variants will get breast cancer, and perhaps even more importantly, those variants are associated with just 3% or so of all breast cancers. It's better perhaps to say that breast cancer can sometimes be one of the body's responses to the complicated shifts in gene regulation caused by the absence of a healthy, functional BRCA1 protein, but that we can find that same response also to many other different perturbations the body might experience. And this is an example of how many diseases seem to be cases of what the developmental biologist Conrad Waddington called canalization, whereby a wide range of circumstances at the molecular level or at the cell level become channeled into the same physiological response. And it's because of this kind of canalization that some diseases, so think of COVID and flu, can become very hard to diagnose and to distinguish from the symptoms alone, because the symptoms are canalized and look very similar. It's also for this reason that drugs can be repurposed, and this has become a big deal, drug repurposing, that a drug developed for one condition might turn out to be effective for another apparently quite different condition. That was the case for dexamethasone, which was developed as an anti-inflammatory for rheumatoid and allergy conditions, but turned out also to be effective against COVID. So the real question there is, how does the body get into one of these disease valleys, and how might we get it out? But while there are practical questions like this for wanting to understand how it is that life works, I think we should be interested in that question simply to make us appreciate all the more how astonishing life is. The idea that what makes living matter alive, what makes it different from a rock, the idea that it's like that because it's simply been pre-programmed to be alive, doesn't really tell us anything. And the idea that an account of life is to be found by gradually increasing the magnification until we arrive at molecules is a fantasy, as the Nobel laureate Albert Szent-Györgyi understood very well when he said this: "My own scientific career was a descent from higher to lower dimension, led by a desire to understand life. I went from animals to cells, from cells to bacteria, from bacteria to molecules. On my way, life ran out between my fingers." What this comes down to then is looking for an explanation of how life works that really does justice to the truly amazing nature of life itself. Saying that life is a machine made by genes or like a computer running a program is not only wrong from the perspective of what modern biology tells us, it banishes life from biology altogether. In the way that words like agency and purpose and meaning are often almost taboo in biology, I sense a kind of embarrassment at how extraordinary life really is. Here we are, pieces of the universe that have become aware of themselves. That sounds poetic, but it's simply the literal truth. Biologists should never forget that that's really what they're trying to understand and explain. And so no wonder it's hard, but I think we should refuse to accept too cheap, too mechanical, and too reductive an answer to this question. I think the most profound question in science, how life works. Thank you.